Hello everyone, this is Science Minds and you're watching the audiovisual chapter of class 11th biology. This is the second part of chapter 6, anatomy of flowering plants, and we are starting off with the tissue system. We are discussing types of tissues based on type of cell present. Let us now consider how tissues vary depending on their location in the plant body. Their structure and function would also be dependent on location. On the basis of their structure and location, there are three types of tissue system. There are epidermal tissue system, the ground or fundamental tissue system, and the vascular or conducting tissue system. First, we are talking about the epidermal tissue system. The epidermal tissue system forms the outermost covering of the whole plant body and comprises the epidermal cells, stomata and the epidermal appendages, the trichomes and hairs. The epidermis is the outermost layer of the primary plant body. It is made up of elongated, compactly arranged cells which form a continuous layer. Epidermis is usually single layered. Epidermal cells are parenchymatous with a small amount of cytoplasm lining the cell wall and a large vacuole. The outside of the epidermis is often covered with a waxy thick layer called the cuticle, which prevents the loss of water. Cuticle is absent in roots. Stomata are structures present in the epidermis of leaves. Stomata regulate the process of transpiration and gaseous exchange. Each stoma is composed of two bean-shaped cells known as guard cells, which enclose stomatal pore. In grasses, the guard cells are dumbbell-shaped. The outer walls of the guard cells, away from the stomatal pore, are thin and the inner walls towards the stomatal pore are highly thickened. The guard cells possess chloroplast and regulate the opening and closing of stomata. Sometimes a few epidermal cells in the vicinity of the guard cells become specialized in their shape and size and are known as subsidiary cells. The stomatal aperture or the guard cells and the surrounding subsidiary cells are together called stomatal apparatus. Figure 6.4 is a diagrammatic representation. A is a stomata with the bean-shaped guard cells. B is a stomata with the dumbbell-shaped guard cells. The cells of epidermis bear a number of hairs. The root hairs are unicellular elongations of the epidermal cells and help absorb water and minerals from the soil. On the stem, the epidermal hairs are called trichomes. The trichomes in the shoot system are usually multicellular. They may be branched or unbranched and soft or stiff. They may even be secretory. The trichomes help in preventing water loss due to transpiration. Now let's talk about the ground tissue system or the fundamental tissue system. All tissues except epidermis and vascular bundles constitute the ground tissue. It consists of simple tissues such as parenchyma, collenchyma and sclerenchyma. Parenchymatous cells are usually present in cortex, pericycle, pith and medullary rays, in the primary stems and roots. In leaves, the ground tissue consists of thin-walled chloroplast containing cells and is called mesophyll. Now let's talk about the vascular tissue system or the conducting tissue system. The vascular system consists of complex tissues, the phloem and the xylem. The xylem and phloem together constitute vascular bundles. In dicotyledonous stems, cambium is present between the phloem and xylem. Such vascular bundles, because of the presence of cambium, possess the ability to form secondary xylem and phloem tissues and are hence called open vascular bundles. In monocotyledons, the vascular bundles have no cambium present in them. Hence, since they do not form secondary tissues, they are referred to as closed. When xylem and phloem within a vascular bundle are arranged in an alternate manner along the different radii, the arrangement is called radial, such as in roots. In conjoint type of vascular bundles, the xylem and phloem are jointly situated along the same radius of vascular bundles. Such vascular bundles are common in stems and leaves. The conjoint vascular bundles usually have the phloem located only on the outer side of the xylem. Now figure 6.5, various type of vascular bundles. A is a type of a radial vascular bundles. B is a kind of a conjoint or the closed one. C is a type of a conjoint open type. Now let's talk about the anatomy of a dicotyledonous and monocotyledonous plants. For a better understanding of a tissue organization of a roots, stems and leaves, it is convenient to study the transverse section of mature zones of these organs. First we are talking about the dicotyledonous root. Look at the figure 6.6a. It shows the transverse section of a sunflower root. The internal tissue organization is as follows. So A is a dicot root and actually a transverse section of a dicot root in which there are root hairs which are protruding from the epidermis or the epiblemma, and these are unicellular. Then comes the cortex, which is several layers of parenchyma cells. Then comes the endodermis, and most importantly the endodermis is covered with suberin or a waxy material in a form of a casparin strip, which is actually transitional and tangential as well as radial walls of the endodermal cells. And then comes the pericycle. Pericycle is also consists of parenchymatous cells. Then comes the pith, which is small and inconspicuous. Parenchymatous cells which lie between the xylem and the phloem, okay, which is between a xylem and phloem, are called conjunctive tissue. So let's talk about the paragraph now. The outermost layer is epiblemma. Many of the cells of epiblemma protrude in the form of unicellular root hairs. The cortex consists of several layers of thin-walled parenchyma cells with intercellular spaces. The innermost layer of cortex is called endodermis. It comprises of single layer of barrel-shaped cells without any intercellular spaces. The tangential as well as radial walls of the endodermal cells have a deposition of water impermeable waxy material suberin in the form of casparin strips. Next to endodermis lies a few layers of thick-walled parenchymatous cells referred to as pericycle. Initiation of lateral roots and vascular cambium during the secondary growth takes place in these cells. The pith is small or inconspicuous. The parenchymatous cells which lie between the xylem and the phloem are called conjunctive tissue. There are usually 2 to 4 xylem and phloem patches. Later, a cambium ring develops between the xylem and the phloem. All tissues on the inner side of the endodermis, such as pericycle, vascular bundles, and pith, constitute the stele. Now let's talk about the monocotyledonous root. The anatomy of a monocotyledonous root is similar to the dicot root in many respects. Figure is 6.6b, which is a transverse section of a monocot root. It has epidermis, cortex, endodermis, pericycle, vascular bundles, and pith. As compared to the dicot root, which have fewer xylem bundles, there are usually more than 6 or polyarch xylem bundles in the monocot root. Pith is large and well developed. Monocotyledonous roots do not undergo any secondary growth. So here we end the second part of the chapter 6, Anatomy of Flowering Plants. To watch more such videos, do subscribe the channel and press the bell icon so you don't miss any video. And if you want us to create more such videos on any other book or subject, do mention in the comment section please. Like and share the video to keep us motivated to create such content. Thank you for watching the video. Bye bye. Love.